This lecture on glycoproteins represents just a small subset of the total universality of this type of molecule, glycoproteins, that are found all over the human body as well as in the plant and animal kingdom. So first, let's just define a glycoprotein. Glyco means some sort of sugar or an ensemble of sugars attached to the protein. So I just listed two examples of glycoproteins. They're found often in dental plaques. So this is a community of bacteria that usually secrete a glycoproteinaceous substance, protein conjugated to a sugar, and you actually see a consistency that is very conducive to what we find in plaques. So a lot of dental plaques are found by bacteria that secrete these substances. A lot of them are enriched in glycoproteins, different types of glycoproteins. Also in medical devices, another practical application that is adversarial to the community is that if you take a stent or any type of catheter tube that goes or inserts into the body, a lot of those catheters can be susceptible to infection by polymicrobial communities, and those polymicrobial communities adhere to these surfaces, whether it's a catheter tube or a hard surface like tooth enamel. They adhere to it by secreting a very, very sticky, waxy substance known as glycoproteins. So that's just one application, but the applications just span farther than that. Glycoproteins usually are going to be on the surface, outside surface of the cell, and there will be receptor attachment sites. So again, this harkens to this universality of this huge molecule, a huge database of molecules that we can describe, lots and lots of practical applications. So this is just a review of what we will be talking about. Again, remember that glycoproteins represent sugar plus proteins. We're also going to step up one level of complexity later on in this lecture and talk about proteoglycans, which are glycosaminoglycans conjugated to proteins. Don't forget your glycosaminoglycans that we mentioned in the previous lecture, such as heparin, though it's not really involved in the extracellular matrix. That's involved in preventing blood coagulation or blood thinning. But the other ones, such as chondroitin 6-sulfate, chondroitin 4-sulfate, hyaluronic acid, these are going to really maintain the extracellular matrix, and we add that to proteins, we get a proteoglycan. Bacterial cell walls, our second bullet point here, are also a glycoproteinaceous substance, and that is really the basis for discriminating between a gram-positive cell and a gram-negative cell. And the third bullet point talks about just more practical applications of glycoproteins. In eukaryotes, glycoproteins often play a role as receptors or receptor recognition sites. So let's first talk about proteoglycans. Again, the main topic of this lecture is sugars attached to proteins. We learned about proteins in Module 1, and now we're learning about sugars and how they bind through glycosidic bonds, and this being Module 2. So proteoglycans are another layer of complexity to help maintain the viscosity and aqueous matrix nature of the extracellular matrix. In a proteoglycan, you actually have a chain of a glycosaminoglycan. And that glycosaminoglycan can be a main chain of chondroitin sulfate, it could be a main chain of hyaluronate, whatever it may be, it's just a main chain. And attached to those sugars, there's going to be a branch point, and that branch point could also be another set of glycosaminoglycans, such as chondroitin sulfate or hyaluronic acid. Now, what's shown here is actually the proteinaceous portion of the proteoglycan. These proteins actually branch out, and that's your protein with your peptide bonds, and then attached to those amino acid side chains are sugars. Now, the important point of this class is that there are two ways you can conjugate a sugar to a protein. You can do it through N-linked glycosylation, so this would be N-linked oligosaccharides, or you can have O-linked glycosylation. N-linked glycosylation is attaching a series of sugars to the nitrogen of asparagine, And O-linked glycosylation is attaching a series of sugars to the oxygen of serine. If you look at a proteoglycan as a whole, under electron micrograms, it actually looks like a bottle brush. So many times we call this the bottle brush model. It's a model that helps us distinguish how a proteoglycan looks like. So you can actually see here, here's your main chain glycosaminoglycan, and these extension outwards is your protein, and extended from the protein are going to be more additional sugars, either N-linked or O-linked. So again, this is found extensively in cartilage, but it's also found as a major maintenance of the extracellular matrix. Remember, the extracellular matrix must have viscosity, must have flow, and be resistant to shearing. And it's this jelly-like matrix material, proteoglycans, part played by the glycosaminoglycan network, actually help in facilitating the matrix aqueous jelly-like consistency that's required in your joints and your cartilage. Also the tendons of your eye, blood vessels. Think of the elasticity. Proteoglycans play an important role in that, and it's a glycoprotein in the sense that you have proteins attached to those proteins, sugars. There are thousands of examples of glycoproteins. In fact, there's entire courses in some schools that dedicate it just to glycoproteins. So some examples of glycoproteins are the proteins that HIV binds to the target cell. In this case, the target cell for the HIV are T cells, the immune system cells. Collagens, we talked about the 4 different types of collagens that are found in the body. It's one of the more abundant, specialized structural proteins in the human body. Collagens will interact tremendously with glycoproteins. Thyroid-stimulating hormone is an example of a glycoprotein, a protein that has a sugar or several sugar moieties attached. Fertilization between a sperm and an egg, the generation of a zygote. That fertilization event is met by glycoproteins recognizing each other. A large class of proteins known as mucins are high molecular weight glycoproteins. That seems to be upregulated in certain cancers. So a lot of normal cells, as well as cancer cells, seem to have make these glycoproteins, probably for adherence and sticking to substances. Remember, go back to the first slide and look at biofilms and look at the biofilms made on catheters. These are naturally going to be very sticky and viscous, and so that allows for adherence to surfaces. Other types of glycoproteins, this is just a small list by the way, is transferrin. That's an iron binding protein in the cell. So a lot of the proteins in the cell are going to be glycoproteins. Also, a lot of receptors in the cell, more often than not, will be glycosylated. Again, how are these sugars added? They are added either through the N-nitrogen of asparagine, its side chain, that's called N-glycosylation, or they could be added through the oxygen atom of a serine or threonine amino acid side chain. In that case, that's O-linked glycosylation. N-linked glycosylation usually occurs in the endoplasmic reticulum, whereas O-linked glycosylation occurs in the Golgi apparatus. So let's take a look at N-linked glycosylation. If a protein that's going to emanate from the ribosome will become its ultimate fate to being a glycoprotein, it's going to happen in the geographic location of the endoplasmic reticulum. That is the cellular location. And the N-linked glycosylation event occurs in what is known as a co-translational manner, meaning as the protein emanates from the ribosome, as the peptide bond is being catalyzed, it's being done in the rough ER and in the lumen of the rough ER. And as amino acids are funneling out through the ribosome, they get attached a core set of sugars. So N-linked glycosylation, which we mentioned before, occurs in the lumen of the rough ER, is going to have this saccharide core, and it gets trimmed. So inside the lumen of the ER, we have a lot of endoglucosidases. They actually are going to add sugar residues, and we also have exoglucosidases, which will trim and edit. So you notice here that at the beginning step, you add a 14 residue oligosaccharide. It gets trimmed. It goes through several what we call editing, and the sugars get edited ultimately to the final glycosylated product. Most of the editing and remodeling of the final glycosylated protein product, it will occur within the lumen of the endoplasmic reticulum. Also note the types of sugars that are added, such as fucrose, sialic acid, glucose, galactose. Notice we have one L sugar, so L sugars are going to be common. And then we also have a combination of different types of sugars. Some of these are derivatized, such as N-acetylglucosamine, that's an aminated sugar. And then some of these are just regular epimers or isomers of glucose. So there's a lot of variety in the sugars that are added. Functionally speaking, the glycoprotein, that sugar residue on the nitrogen attachment site may serve as a receptor attachment. It provides a lot of conformational flexibility to the glycoprotein, and overall it can be used mostly as a receptor site, and it's going to be important for trafficking. So if this is going to be a plasma membrane protein, these oligosaccharides will traffic it to the plasma membrane. If it's a protein destined for the lysosome, then these sugars will traffic that to its final destination, the lysosome. So the specific signature of these sugars also determine its cellular destination. The overall point of this slide is that you're going to have a lot of editing and trimming of sugars. This is end-linked glycosylation, where oligosaccharides are added to the nitrogen side chain of asparagine. Ultimately, after editing, you'll have a pentasaccharide core. So this goes back to our review of sugars. Here you have the alpha anomer of mannose. It forms a 1,6 glycosidic bond with the number 6 carbon of this mannose. Here we have the alpha anomer of mannose. It's forming a 1,3 glycosidic bond with carbon number 3 of mannose. Here's the beta anomer. Its carbon is forming a bond with carbon number 4 of N-acetylglucosamine. And that beta anomer, carbon number 1, is forming a bond with carbon number 4 of N-acetylglucosamine. So amongst all the editing, you will get this pentasaccharide core structure, and further trimming and further processing is added. Remember, this is occurring within the confines of the endoplasmic reticulum. We have a series of modeling and editing and addition and subtraction of these sugars from the pentasaccharide core. If you look at where this sugar is attached, again, it's important to recognize that end-linked glycosylation occurs via the nitrogen of the side chain of asparagine. So here's your peptide backbone, and you can see sort of a consensus sequence. The various enzymes that will add the sugar, the endoglycosidases or the exoglycosidases, which will edit them, usually recognize, generically speaking, a serine or threonine, any amino acid X, and then the nitrogen of the asparagine, which will be the target site for glycosylation. Again, glycosylation occurs in the ER, and it gets trimmed in the ER. Also, editing occurs in the Golgi apparatus. O-linked glycosylation also occurs in the Golgi apparatus. So here we have O-linked glycosylation. Again, look at the oxygen of serine or threonine. It's the site. And we really don't have a core structure here that we do with N-linked glycosylation. If you remember that pentasaccharide core, we do see a little bit of consensus sugar with beta-galactose, which its anomeric carbon forms a bond with carbon number 3 of N-acetylgalactosamine attached to the oxygen of either a serine or threonine. Remember what these sugars are going to do. The addition of these sugars to these proteins give it more conformational flexibility, conformational freedom. They impart a charge. They make a substance more viscous if it's going to be a proteoglycan. And again, receptor attachment sites. So a lot of proteins get decorated, and they get decorated with a lot of sugars, either N-linked or O-linked. Now let's go to the second portion of this lecture, and that is a major glycoprotein that's important in the microbiology community, and that is peptidoglycan. Peptidoglycan actually is a merger of two words, peptide and glycan. And glycan is just an abbreviation for the word sugar. So here's peptidoglycan in a gram-positive cell. You notice that it's very thick, and gram-positive bacteria, such as your Clostridium difficile or your Clostridium genus, they're going to have a very thick peptidoglycan layer, so they're going to exclude the secondary stain if you perform a gram stain on these. If you contrast this with gram-negative bacteria, the classic example is E. coli and all of the Enterobacteriaceae, that family of gram-negative bacteria. They have a very thin peptidoglycan, and supplanted on top of that cell wall is an outer membrane. For E. coli, that would be lipopolysaccharide and a couple of other components. So this is actually taken up by the secondary stain during gram staining, and that is why these things have a differential staining, because the thick peptidoglycan, that cell wall in gram-positive, excludes the secondary stain, while that gram-negative bacteria, it picks up that secondary stain because you have a very thin peptidoglycan cell wall built upon that outer membrane components. So let's talk about how peptidoglycan looks like. Let's talk about its structure. And here it is. This is the structure of peptidoglycan. It's important to recognize what it is a polymer of. It's a polymer of N-acetylglucosamine, forming a beta-1,4 glycosidic bond with N-acetylmuramic acid. Once again, peptidoglycan is a disaccharide polymer of N-acetylglucosamine. Its beta anomer forms a glycosidic bond with carbon number 4 of N-acetylmuramic acid. So a disaccharide polymer of N-acetylglucosamine and a beta-1,4 glycosidic bond with N-acetylmuramic acid. And that disaccharide polymer continues onward. Now that's the glycan component. The peptide component is right here. You actually see 5 amino acids. One is L-alanine. There's that L-amino acid. We have an isoglutamine forming an isopeptide bond with lysine. So the peptide bond is with the side chain, not with the main chain. So a side chain peptide bond is called an isopeptide bond. This isoglutamine forms an isopeptide bond with lysine. And then we have a D-alanine. Notice the mixture of D and L amino acids. And that peptide is attached to, or the substituent on carbon number 3. So we have here we have a tetrapeptide attached to N-acetylmuramic acid on this disaccharide polymer. So that's not all, because this is one strand of peptidoglycan. And how do we connect adjacent strands? We connect adjacent strands with a pentaglycine bridge. A pentaglycine bridge is 5 glycine residues. So the pentaglycine bridge actually bridges together each strand of the peptidoglycan disaccharide polymer. Right? So here's your tetrapeptide that you have seen before, and then attached here to the third amino acid of that tetrapeptide, that is a lysine, on one strand. Here you have 5 glycine. Glycine 1, glycine 2, glycine 3, glycine 4, glycine 5, and that's going to form a covalent bond with the 4th amino acid in the tetrapeptide, and that is an alanine. So this is how the connectivity occurs through adjacent strands. The connectivity occurs through that pentaglycine bridge. Pentaglycine bridge, which connects really the tetrapeptide components of the disaccharide polymer. So don't forget the tetrapeptide is attached to N-acetylmuramic acid. The disaccharide polymer consists of N-acetylglucosamine with a beta-1,4 glycosidic bond to N-acetylmuramic acid, and that's actually one strand. To join adjacent strands, you have 5 glycines, the pentaglycine bridge as it is called. So this rigidity from that pentaglycine bridge connection, as well as its overall 3-dimensional structure, allows this to wrap around a cell wall. So here's a cartoon model of peptidoglycan wrapping around gram-positive bacteria, and this would be a very thick peptidoglycan layer. We would expect a very thin peptidoglycan layer for gram-negatives. So the model of this is just that this, if I could go back to the previous slide, this just sort of wraps around and conforms to the shape of the particular gram-positive or gram-negative bacteria. If it's gram-positive, you have more wrapping, and more of a thicker layer. If it's gram-negative, you have less wrapping, and you have the lipopolysaccharide and outer membrane constituents interacting with that. So this is sort of the cell wall component of gram-positive and gram-negative bacteria. That cell wall gives it some sort of rigidity. It excludes it from the outside environment. It provides for isotonic, or so that nothing leaks in or nothing leaks out inside the cell. And that is basically one of the functions of peptidoglycan. It's a cell wall, but more importantly in the context of this discussion, it's a glycoprotein. It's a very complicated glycoprotein, but it's a glycoprotein. One thing about peptidoglycan, it's a target site for this, one of the more famous antibiotics that was discovered, and that's penicillin. Penicillin is a beta-lactam antibiotic, so this is the beta-lactam ring. And it goes in and it inhibits peptidoglycan biosynthesis. It inhibits the cross-linking step where peptidoglycan can cross-link to itself so that it could have that thick, rigid layer. And so penicillin was actually discovered by Alexander Fleming, and he discovered it basically by accident. He found that this was a substance secreted by fungus or mold, and he left the petri dish out at the windowsill. That's an absolute cardinal no-no in microbiology, leaving the agar plate open. But by accident he did that, and by his fortuitiveness, he was able to find when he came back that the agar plate had zones of inhibition, and he isolated that substance secreted from the mold that killed the bacteria. It was penicillin. Most bacteria now have a penicillinase that can actually cleave the beta-lactam bond, can cleave this ring, and disintegrate penicillin. So a lot of bacteria, one method to develop resistance is to encode for a penicillinase, and a lot of bacteria that are resistant to penicillin have a penicillinase. Coming back away from bacteria, let's talk about eukaryotes for a second. N-linked and O-linked glycosylation is the primary method for getting sugars into a eukaryotic protein, and some of the functionalities of which are just additional degrees of motion, additional conformational flexibility. In eukaryotes, glycoproteins generally are receptor sites, and you can see here's the protein in a space-filling model, and here is in stick figures all the different conformations that the sugar molecule or that ensemble can take. You can see here more freedom, and it can explore more degrees of space. And three-dimensionally speaking, if there's a receptor or any binding target for this molecule, the binding target has a lot more freedom and has a lot more area of access. So that's why we tend to think that glycoproteins make very good receptors, very good specificity sites. If you remember, HIV actually binds to T cells via a glycoprotein, GP60 and GP20, glycoprotein 60 and glycoprotein 120. Another important aspect of these glycoproteins is that they are on the surface of red blood cells, and they are the determinants of the A, B, and H blood type. So dependent on what type of sugars are attached to the red blood cell will depend on what type of blood type you are, and also will depend on what type of antibody the body produces. So we have H-type blood, which has this pair of sugars. Notice we have an L sugar with an alpha-1 glycosidic bond to carbon number 2 of galactose. Its beta form forms an glycosidic bond with carbon number 4 of N-acetylglucosamine, and that unit is the surface antigen for H-type blood cell. We also have a surface antigen for A-type and a surface antigen for B-type. So these oligosaccharides not only provide conformational flexibility for glycoproteins, but they also play an important role on whether or not you can accept a specific type of blood transfusion, because again, it's these sugars attached to proteins on the site of red blood cells that determine your specificity for blood. So that is a quick synopsis of glycoproteins. It's a very universally regarded macromolecule, and it's everywhere. Okay, so proteins by themselves are important. Sugars by themselves are diverse and important, but when you join them together and get a glycoprotein, you have tremendous amount of functionality and you have a tremendous amount of flexibility in terms of its function.